Assalamualaikum warahmatullahi wabarakatuh. Good day, everyone. My name is Oloan Adikara Lumban Tobing, and I'm honored to present my thesis proposal titled In silico analysis of mangiferin derivatives for inhibition of the WNT beta-catenin pathway to prevent carcinogenesis in hepatocellular carcinoma using molecular docking. This research is conducted under the supervision of Professor Dr. Harno Dwi Pranowo and Professor Trijo Koraharjo. Our goal is to explore the potential of mangiferin derivatives as inhibitors of the WNT beta-catenin pathway, which plays a crucial role in the development of HCC, one of the deadliest forms of liver cancer. Introduction. Liver cancer, particularly HCC, is a major health issue, with significant mortality and morbidity worldwide. The global incidence rate of liver cancer is approximately 12.4 per 100,000, with a mortality rate of 7.6 per 100,000. In Indonesia, this situation is even more concerning, with PHPI reporting an age-standardized incidence rate of 13.4 per 100,000 people. This high incidence, coupled with the aggressive nature of HCC, underscores the importance of finding new treatments. Recent studies have highlighted the potential of natural compounds such as mangiferin in inhibiting the WNT beta-catenin pathway, a key driver of HCC carcinogenesis. This pathway's inhibition could offer a novel approach to preventing or treating HCC. Now, as you can see here, the symptoms of liver cancer, more specifically HCC itself. We have thoracic radiculopathy, which will in turn affect the human body with neuropathic pain. You also experience enlarged liver and also jaundice. Now, one of the possible applications for new researchers involved in HCC treatment is using mangiferin, which is derived from the bark, the leaf, and the stem of Mangifera indica or mango. Now in this study, we will examine 5 specific derivatives of mangiferin: homomangiferin, isomangiferin, neomangiferin, mangiferin 6-galate, and also noratheriol. These derivatives were selected for their structural variations, which may confer enhanced biological activity compared to mangiferin itself. For example, homomangiferin has an additional hydroxyl group, which could potentially increase its ability to form hydrogen bonds with target proteins. Isomangiferin and neomangiferin have different glycosylation patterns, which might affect their solubility and interaction with proteins. Mangiferin 6-galate is a galoylated derivative that may exhibit stronger antioxidant properties, while noratheriol, which lacks the glucose moiety, might offer better penetration into cells. By studying these derivatives, we aim to identify which modifications enhance the ability of mangiferin to inhibit the WNT beta-catenin pathway. Now in this slide, we delve into the rationale behind selecting specific receptors for docking with mangiferin derivatives. Each of the receptors plays a crucial role in the WNT beta-catenin signaling pathway, which is implicated in the development and progression of HCC. Starting with WT1, represented here by the PDB code 6B0O, this protein is known for its role in oncogenic gene regulation. By docking mangiferin derivatives with WT1, we aim to inhibit its ability to promote cancerous gene expression, potentially reducing the proliferation of HCC cells. Now moving to LEF1, represented by the PDB code 3OUX. LEF1 is a transcription factor that forms a complex with beta-catenin to activate WNT target genes. Docking mangiferin derivatives with LEF1 is intended to block this complex formation, thereby interrupting the WNT signaling pathway and its contribution to tumor growth. Next, we have FZD7, shown here with the PDB code 6NE2. FZD7 is a receptor that initiates WNT signaling. By impeding the signaling initiation of this receptor, we can potentially prevent the downstream activation of oncogenic pathways, making it a crucial target in HCC therapy. Lastly, CTNNB1, represented by the PDB code 1JDH, encodes beta-catenin itself, a key mediator in the WNT signaling pathway. By disrupting beta-catenin's role through targeted docking with mangiferin derivatives, we aim to hinder its ability to sustain WNT signaling, which is vital for cancer cell survival and also proliferation. Together, these docking studies are expected to demonstrate how mangiferin derivatives can strategically inhibit various components of the WNT beta-catenin pathway, offering a multifaceted approach to preventing and potentially treating HCC. Here are the research objectives. The first one is to determine the binding affinities of mangiferin derivatives to key WNT beta-catenin pathway components, including beta-catenin, TCF, LF, Frizzle7, and also Wilms tumor 1. The second one is to also conduct molecular dynamic simulations to assess the stability and dynamics of mangiferin derivatives bound to WNT beta-catenin pathway components, identifying conformational changes or interactions that could influence their inhibitory potential. For the benefits, this study will provide insights into how these substances can effectively inhibit the activation of specific pathways, contributing to the development of more targeted and potent inhibitors for the treatment or prevention of HCC. The results may contribute to the creation of novel pharmacological treatments that lower the risk of HCC and maybe other malignancies linked to the dysregulation of the WNT beta-catenin pathway. Now we shall continue to the theoretical background. So first we're going to discuss about the WNT beta-catenin pathway. Now, this is a very crucial signaling pathway involved in various cellular processes, including cell proliferation, differentiation, and also migration. Under normal conditions, this pathway is tightly regulated, but its dysregulation is a common feature in many cancers, including HCC. The pathway is initiated when WNT proteins bind to receptor complexes on the cell surface, composed of frizzle proteins and also LRP56 co-receptors. This binding inhibits the activity of the destruction complex, which normally keeps beta-catenin levels low by promoting its degradation. When the destruction complex is inhibited, beta-catenin accumulates in the cytoplasm and eventually translocates to the nucleus, where it activates the transcription of target genes involved in cell proliferation and survival. In HCC, aberrant activation of the WNT beta-catenin pathway leads to uncontrolled cell growth and tumor development. Therefore, targeting this pathway could be an effective strategy for inhibiting HCC progression. Now, HCC itself is the most common type of liver cancer, accounting for more than 80% of liver cancer cases. The pathogenesis of HCC is complex and involves multiple genetic and environmental factors. One of the key drivers of HCC is the abnormal activation of the WNT beta-catenin pathway, which leads to increased cell proliferation, invasion, and metastasis. The activation of this pathway is associated with poor prognosis and resistance to conventional therapies. Understanding the molecular mechanisms underlying HCC and identifying novel therapeutic targets are essential for developing more effective treatments. This study aims to investigate how mangiferin derivatives can inhibit the WNT beta-catenin pathway, thereby preventing or reducing the risk of HCC development. Mangiferin itself is a natural polyphenol found in Mangifera indica, commonly known as mangoes. Its chemical structure consists of a xanthone core attached to a glucose molecule, which enhances its solubility and biological activity. Mangiferin has been extensively studied for its diverse pharmacological properties, including its anti-inflammatory, antioxidant, anti-cancer, antiviral, neuroprotective, and anti-diabetic effects. In the context of cancer, mangiferin has shown promise in inhibiting various signaling pathways involved in tumor growth and metastasis. The focus of this study is to explore how mangiferin, and more specifically its derivatives, can inhibit the WNT beta-catenin pathway in HCC. Now let's delve deeper into the specific mangiferin derivatives studied in this research. Homomangiferin, with its additional hydroxyl group, is hypothesized to form stronger hydrogen bonds with target proteins, potentially enhancing its inhibitory effect. Isomangiferin and neomangiferin, which differ in the position of their glycosylation, may have distinct solubility profiles that could affect their bioavailability and interaction with the WNT beta-catenin pathway. Mangiferin 6-galat, a derivative with the galloyl group, is known for its potent antioxidant properties, which may contribute to its anti-cancer effects by reducing oxidative stress within the tumor microenvironment. Noratheriol, which lacks the glucose moiety, is expected to penetrate cell membranes more easily, potentially increasing its efficacy as a WNT beta-catenin pathway inhibitor. And by comparing these derivatives, we aim to identify which structural features are most beneficial for inhibiting HCC. Now, the key receptors targeted in this study are WT1, Wilms tumor 1, LEF1, lymphoid enhancer binding factor 1, FZD7 or frizzle 7, and CTNNB1 or beta-catenin. WT1 is a transcription factor with dual roles, acting as both a tumor suppressor and an oncogene, depending on the context. Now, in HCC, WT1 is often overexpressed and contributes to tumorigenesis. LEF1 is another transcription factor that works in conjunction with beta-catenin to regulate the expression of target genes involved in cell proliferation and survival. FZD7 is a receptor that initiates the WNT signaling cascade by binding the WNT proteins, leading to the activation of the WNT beta-catenin pathway. Now finally, catenin beta-1 encodes beta-catenin, the central player in the WNT beta-catenin pathway. Inhibiting these receptors could disrupt the WNT beta-catenin signaling, thereby preventing HCC progression. And what is molecular docking? So, molecular docking is a computational technique used to predict the binding affinity and orientation of a ligand, such as mangiferin derivatives, within the binding site of a target protein. Now in this study, I will proceed to use a YASARA software to perform molecular docking simulations, which will help us understand how these derivatives interact with the key receptors involved in the WNT beta-catenin pathway. The docking process involves generating multiple possible configurations of the ligand-receptor complex and scoring them based on their predicted binding affinity. The top scoring configurations will provide insights into the potential inhibitory effects of the mangiferin derivatives of the WNT beta-catenin pathway. This step is crucial for identifying which derivatives have the strongest interactions with the target proteins, making them the most promising candidates for further investigation. Now we shall continue to the research hypotheses. Now, based on the current literature and preliminary data, I formulated two main hypotheses for this study. The first hypothesis is that mangiferin derivatives will exhibit higher binding affinity and specificity for key proteins in the WNT beta-catenin pathway, such as beta-catenin, LEF1, and frizzl7, compared to mangiferin itself. This increased binding affinity and specificity are expected to result in a greater inhibitory potential against pathway activation and cancer progression. The second hypothesis is that these derivatives will demonstrate enhanced conformational stability and favorable interactions within the binding sites of WNT beta-catenin pathway components during molecular dynamic simulations. This stability and interaction profile will likely lead to more effective inhibition of the pathway, thereby reducing the risk of HCC development. Testing these hypotheses will provide valuable insights into the potential of mangiferin derivatives as therapeutic agents for HCC. Now, the research methodology. Now, the research methodology is divided into two main phases: molecular docking and molecular dynamic simulations. In the molecular docking phase, the ligands, which are the mangiferin derivatives, will be prepared and their energy minimized to ensure they are in their most stable form. The receptors, which include WT1, LEF1, frizzl7, and catenin beta-1, will also be prepared. The docking simulations will be performed using the Yasara software with specific parameters such as temperature of 298K and an exhaustiveness of 25. The docking will be run multiple times, and the scoring function will be Amber, a force field specifically designed for docking studies. The two complexes with the best binding affinities will be selected for further analysis. This phase is crucial for identifying which mangiferin derivatives have the strongest potential to inhibit the WNT beta-catenin pathway. Now, the second phase of the research methodology involves molecular dynamic simulations. In this phase, the selected ligand-receptor complexes from the docking phase will be subjected to molecular dynamic simulations to assess their stability and interactions over time. The simulations will be run at temperature of 298K and a pressure of 1 atm using the Amber 14 force field. The boundary conditions will be periodic, and the simulation will be run for a duration of 100,000 picoseconds or 100 nanoseconds. During the simulations, various parameters such as RMSD, root mean square deviation, RMSF, root mean square fluctuation, hydrogen bonding, cluster analysis, and energy changes will be monitored. These analyses will help us understand how the mangiferin derivatives interact with the WNT beta-catenin pathway components over time and whether they remain stable and effective inhibitors throughout the simulation period. And that is all from me. Thank you for your attention.